Hello, Dr. Drury. Thank you for joining us here today.
My pleasure.
Well,
Hi,
I'm Pete
David.
Deswick. I'm with the Cure
Dana.
Starts Now and also the DIPG DMG Collaborative. And I appreciate you joining to go ahead and talk a little bit about your project. But before we jump into it, I wanted to let you know, and we'll get this out of the way, that the DIPG DMG Collaborative has decided, along with the advice of our medical advisory, to go ahead and award you a partial grant funding of
That's so exciting.
$50,000,
I love it.
okay, for your project on effectively the open science elements and identifying the ALK2 inhibitors for DIPG.
That's
So,
fantastic. Thank you. This is the most dramatic receipt of a grant ever. I love it.
well, and you know, it gets the pressure out of the way. So now we can actually ask you about the grant
Yeah,
and get a little bit more detail with that. Well, in moving down that direction, I also want to introduce Dan, who has a few words as well.
perfect.
Yes. Hi, Dr. Drury. Dan Weiner. I am a member of the Medical Advisory Council.
Okay.
And on behalf of the Medical Advisory Council for the Cure Starts Now and the members also of the DIPG and DMG Collaborative, we really want to offer our appreciation for the work you do and are planning to do with the grant. Thank you.
You're welcome. Thank you.
Hello, my name is Ken Oman. My wife and I started the chapter of the Cure Starts Now in memory of our daughter Selena, who passed away 10 of DIPG, and on behalf of the 52 chapters of the DIPG Collaborative and the Cure Starts Now, I just want to say thank you very much for the research that you do and helping to find a cure for this terrible disease.
You're very welcome, and I'm excited to be a part of the team, and I can't wait to help.
Well, and now let's talk a little bit about the grant, if we can. You know, this is a little different because number one, of the open science element of it, but also the potential of the ALK2 inhibitors. Can you talk a little bit about you know, what that has the potential to do, and then also about how this is different than some of the other projects out there?
Yeah, I think there are a couple elements that are different, and it's really the driver is based on, I think, well, there are two drivers. One driver is, as a rare pediatric cancer, the business case for investment by big pharma is poor, right? And for where there's no profit motive, if a company decides to get into this, what's likely going to happen is if they're successful, they're going to have to charge a lot, and it's going to be unreachable for most families. So the idea behind M4K is to basically remove that profit motive and say, let's do this. How can we do this? How can we rejigger the discovery paradigm and make this open science collaborative and sort of community driven, with the express goal of identifying effective medicines that are cost effective. And so sort of remove the profit motive and say, this is for the common good. How can we redo this? And so basically the people at M4K who are sort of more business savvy than I am have come, well, we've learned that sharing and pre-competitive, you can actually get people to contribute in kind. And so all that, those add up as real contributions. But down the road, it's sort of this idea of, without patents, you can get regulatory exclusivity from the FDA, and that has some value that can be leveraged by investors in certain ways that I'm not probably really equipped to talk about. And through that and these priority review vouchers, which become available, there are ways to incentivize other investment to help get along the way. But the bottom line for M4K and this sort of global team is, we don't want financial incentives to drive it. We want the science to drive it, and think that we can raise enough money through partnerships like you and others to say, let's test this hypothesis in patients, which is where we need to. And that sort of gets to the hypothesis. And from our read of the literature, I ALK2, this kinase seems like a really strong target. So not our labs and other labs have demonstrated through knockdown of the protein and through inhibitors that aren't perfect that it can really affect the growth of DIPG cell lines. And there's in vivo work even showing that compounds can be efficacious. It's of course still a long road, but it feels like a really good target. My angle really has been, I've worked on kinases for many, many years. Kinases are super tractable drug targets. So by tractable, I mean the community has learned that you can, if you identify a kinase of interest for your disease, the medicinal chemists and chemical biologists of the world have become really good at making potent and selective cell active drugs that can be effective against those kinases. And so it's a good target class that people know how to work on. So that's promising. So that sort of marries why ALK2 and why M4K strategy, which is what we want to do. We want to really, over the next 2 years, find a compound that meets all the requirements, and that will start to include safety in animals so that you can get approval for an IND. And we hope over the next 2 years to get all that data and package together and present that to the FDA so that we can embark on clinical trials. Obviously, there's a lot of things between now and then, and a lot of legwork that has to be done in terms of setting up those trials, who is it done with. But those discussions have started with M4K leadership and how do we find the good news is the clinical trialists in this space, they want new medicines, right? They understand the need. And so hopefully through these partnerships, we can get there and run the real experiment, which is, does a potent and selective inhibitor of this kinase, can it be of consequence to these patients?
Yeah. Well, and I think it's that type of innovative modeling and new strategies I think that we need to try to be able to get this to the point that we have the actual cure of it. So we thank you for your work.